Hi, I'm Kim McIntosh, and this lesson is about human genetics. Our objectives here are to talk about Mendel and his experiments that led to his principles of dominance, segregation, and independent assortment. We're going to solve genetic problems that involve monohybrid and dihybrid crosses. We'll talk about the difference between genotype, phenotype, heterozygous, homozygous, dominant, and recessive. We'll talk about the chromosome movement that we learned about in meiosis and how that affects the characteristics of inheritance. We're going to talk about how changes in the chromosomal number can occur and cause some problems. And we will also talk about genetic screening techniques that will tell us if chromosomal abnormalities have occurred. So we're going to start off talking about Gregor Mendel. And Mendel was a monk who did a lot of experiments with pea plants. And he was very curious because he noticed that his pea plants had different traits. And they seemed to have these traits in one variation or another. So if he was looking at seed pods, he noticed that they were either green or yellow. He noticed that they were either constricted or wrinkled or they were full. He noticed the flower color was either purple or white. He noticed that they were either tall or short. And he was very curious about how these traits occurred and how these traits were passed on from one generation to another. And so he did hundreds of experiments with pea plants. Now, what he found out from his experiments was that when he paired a purple plant with a white plant, then he would get all purple offspring. So the white trait would disappear. But then when he would pair those purple plants together, he would get purple plants and white plants again. And then he did this type of experiment with a lot of these different traits, and he found the same thing over and over again. If he paired a yellow seed with a green seed plant, then the next generation, he would have all yellow seeds. But then when he paired those yellow seed plants with a yellow seed plant, in the following generation, he would have some yellow, but he would also have some green coming back. And he was very curious about this. He wanted to know, well, how did the trait, basically one of the traits went away in the next generation, but then would come back again later. And he really wanted to figure out how these traits were passed on, and they could come up in some generations and not come up in other generations. So what we got from these experiments that Mendel did is we got some genetic terminology. And so Mendel came up with the idea of dominant and recessive. So those traits that came up in that second generation, so when he crossed the purple and the white flower plants and he got all purple, he said that purple was dominant. And he thought the white was recessive. Because it came back again later, but it didn't show up in every generation. So he came up with the concept of dominant and recessive. And then also we get genotype and phenotype. So the phenotype is the actual trait that's going to show up in the individual or in the organism. So on this graphic, it shows the earlobes. So you have detached earlobes, and then you have attached earlobes. In Mendel's, the phenotype was the flower color or the seed color, the seed shape. These different traits, what actually shows up in the organism is the phenotype. The genotype is a letter combination that we assign to the trait. So in this case, like here, if we have homozygous dominant, that means that we're going to use a capital letter, and these genotypes are paired. So this would be basically the trait from the mom, and this would be the trait from the dad. And so homozygous means same And dominant means that it's the dominant trait. And so we represent that homozygous dominant with 2 capital letters. And our lettering system, we generally go with the trait that we're looking at. So in this case, the trait is earlobes. And so in this trait, the trait is earlobes. And so the capital E has been used to designate earlobes. Now, over here, with flower color, for the flower color, we might use a capital P because the purple is dominant, and so we would use P. Now, that means for the recessive that would be a lowercase p. So whatever letter we choose for the trait, we're going to stay with the same letter. So we're not going to have a P and a W. We don't want to do that. We want to have a capital P for the dominant trait, and then we're going to have a lowercase p. So our letters are going to be the same. So with this heterozygous condition, what this means is that one of the parents gave the dominant and one of the parents gave the recessive trait. All right, so notice how they use capital E and lowercase e here. We're going to stay with the same letter combinations. And then for homozygous recessive, that means that they're both recessive. So that would be same, and they're both recessive, so we would use 2 lowercase e's. So Mendel came up with this principle of segregation, and what that tells us is that only one of the 2 alleles for a gene is put into a gamete. Okay, and so when the sperm cell is being produced in the father, okay, there is, only one trait or one allele for that trait being put into that sperm cell. So that cell might have the dominant trait, okay, or it could just as easily have the recessive trait. Same situation when the egg cells are being formed. So when the mother is forming an egg cell through that process of meiosis, only one of the 2 alleles is going to go into that gamete. So it could be the dominant trait or it could be the recessive trait. And so that was one of the principles that Mendel came up with, because he figured for the dominant situation to occur, then what was happening was that the mother's trait and the father's trait, so the sperm cell, say it had that dominant allele and it fertilized the egg cell, and say mom's egg cell had the recessive trait, then what would happen in the offspring would be the heterozygous condition. So it would have one allele dominant, one allele recessive, but their phenotype, what actually shows up in that organism, would be the dominant condition, because anytime you have that capital letter there in the genotype, you're going to have the dominant phenotype. So this has led us to do some predictions as far as genetics goes, and we use what's called a Punnett square to make those predictions. And so generally we'll do a Punnett square before there's offspring, or we might even do it after there's offspring to kind of guess as to what the mother and father's genes were. But let's look at how this works. So we do what we call a cross. This cross here is with a father who is heterozygous for a trait and a mother who is homozygous recessive. So this is the cross that we're doing. So this is the heterozygous, the father's trait, and this is the homozygous recessive, the mother's trait. We take these letters and we put them above a 4-square box. So we put one letter over each box. So for the father's genes, we put a capital B over this box, and we put the lowercase b over the other box. For the mothers, we put it over on the side. So we put one letter on the side of these boxes, and we put the other letter on this side. Now, actually, it doesn't even matter whether you put the father's genes on the top or the mother's genes, okay? So it really doesn't matter as long as you put one of their letter combinations on top and the other on the side. Doesn't matter if the father's goes on top or the side, or the mother's goes on top or the side, okay? So let's look at a few different crosses here. Now we're going to do a couple of practice crosses on this monohybrid Punnett square. So let's say that the father is heterozygous and the mother is heterozygous for the trait as well. So for the father, we'll put the two alleles on the top here, like so. And for the mother, we'll put the two alleles along the side. We go through and we put the alleles for the offspring in the box. Now, remember that a Punnett square is a prediction. So this is not trying to say that the mother and father are going to have 4 offspring, but what it's saying is that these are the options for those offspring as to what the genotype and phenotype could be. So when we look at this, we look at a couple of different things. We look at the genotype ratio and we look at the phenotype ratio. So the genotype ratio is to look at how many different letter combinations are there. So in this situation, we have a homozygous dominant, and we only have that in one box. So that's going to be a part of our ratio. We have the heterozygous dominant, and that is in 2 of the boxes. And so that's going to be the next part of our ratio. Then we also have a homozygous recessive here, and that's in 1 box. So our genotype ratio is 1 to 2 to 1. But that's going to be different than the phenotype ratio. So remember, the phenotype is what actually shows up in the offspring. And so in this situation, what we have is this is dominant, this is dominant, and this is dominant. So we have the dominant trait in 3 boxes. And so 3 is the first part of our ratio. We have the recessive trait in 1 box. And so that 1 is going to go there. So our phenotype ratio for a heterozygous times a heterozygous cross, that phenotype ratio is 3 to 1. So this leads us to another principle that Mendel came up with. This principle is called independent assortment. And so Mendel noticed that with all of these different traits that he was looking at, they were independent of each other. And so what that means is that for the pea plants, you could have a round seed or you could have a wrinkled seed. You could also have a yellow seed or you could have a green seed. But those did not go together. They were not linked together. So here you could have a yellow seed that was round, but you could also have a green seed that was round. You could have a yellow seed that was wrinkled, but you could also have a green seed that was wrinkled. So you could get any one of those possibilities. They were not connected to where you had to get a round yellow seed.